Hi, this session is going to focus on getting you acquainted with the basics of using R and OpenMx to analyze twin data. For this session, you may find it helpful to download a couple of files and follow along. Those files are listed here and they can be found at this address. Our major goal for this session is to help you understand some of the basics of analyzing twin data in R and putting together your first twin model for a single phenotype using a basic ACE model. There are several questions that can be answered when you analyze twin data, and they're listed here. Of particular importance for the model we address here are the last two questions, particularly, which sources of variance contribute significantly to the total variance of a trait, and how much of the variation in that trait is due to genetic and environmental influences. While there are other twin and family study designs, for this session, we're going to stay focused on the classical twin study, which uses MZ and DZ twins reared together. The classical twin study has some major assumptions, one of which is that MZ twins share 100% of their genes, and DZ twins on average share about 50% of their genes. Therefore, it's expected that when MZ twin correlations are higher than those of DZ twin correlations, that a phenotype is more similar in MZ twins compared to DZ twins. And when those situations arise, genetic factors are assumed to contribute a portion of the variance to those traits in a population. So as we progress through these slides, I'm hoping to give you a sense of where we're going by providing this roadmap. The first step in this roadmap is to estimate basic summary statistics and visualize those data. And typically that's done by calculating means, variances, covariances, and correlations by zygosity groups. So you see an example here where we take a look at this data set we're working with. This data set is a simulated data set of weight in female adults 65 and older. There are 40,000 twin pairs in this data set with 20,000 MZ twin pairs and 20,000 DZ pairs. When we look at a scatter plot of the value for twin ones versus the values of weights for twin twos in MZ and DZ pairs, you get figures like the ones you have here. Of particular note, if you look at the figure for the DZ pairs, the spread is a bit wider than that of the MZ pairs. Hence, there's greater variation in the DZ twins. Also, we see a lower correlation value in DZ pairs. So now we are building an intuitive sense that from these twin correlations, and we can continue building on those intuitions. So typically there are several patterns that we see when we're looking at twin correlations and comparing MZ and DZ twin pairs. One is like what we see over here in this figure to the left, where the correlation of MZ pairs is exactly twice that of the correlation of DZ pairs. When those situations arise, you can expect that additive genetic influences will play an important role on the variance of that phenotype. When you see the correlations of MZ pairs to be equal to that of DZ pairs, you can expect that the shared environmental influences are likely to be important to influencing the variation of that phenotype. And when you have a situation where the correlation of MZ pairs is larger than that of twice the DZ pair correlation, it's likely that you should expect the influence of additive and dominance effects on the phenotype. Lastly, you might have a situation where the MZ pair correlation is greater than the DZ pair correlation, but it's not exactly twice that of the DZ correlation. In those situations, we expect that additive genetic influences and shared environmental influences are contributing to the variance of that phenotype. And we can begin to calculate the proportions of variance due to genetic and environmental contributions on this outcome using this back of the envelope equations that we have listed here. And we're going to revisit them right now. So as a first step, you can use these back of the envelope calculations, which are often referred to as Falconer equations or Holzinger equations using these formulas. And you can use them to build your intuition around the contribution of additive genetic, shared environmental, and unique environmental effects. So we can go back to the data and now estimate those correlations and visualize them as bar plots in a way that we see right here. And you can begin to estimate using correlations what you think the values of A, C, and E might be, and determine whether or not you would expect an ACE model or an ADE model to best fit the data. In our case, our intuition so far is leading us to the idea that an ACE model would best fit the data. As an aside, the code we've shared with you contains the code that has been used to produce these figures. So if you would like to produce them for yourself or modify them for your own data, you're able to do so. Okay, so great. We now know that additive genetic influences are likely to be important and shared environmental influences are also likely to be important. So let's run an ACE model, right? Wrong. It's always good to continue working through some of those summary statistics. And here we have some of the summary statistics that we calculated through R for the means of twin 1 members of an MZ pair and the twin 2 members of MZ pairs, as well as the variances and covariances between twin 1 and twin 2 members of the MZ pairs. And we've done the same for DZ pairs. We did all of this using the means function, the var function, and cov functions in base R. And that code is identified around line 122 of your script. When you eyeball these results, you might say that these numbers, the means between twin 1 and twin 2, look very similar. And the means for MZ pairs and DZ pairs, they also seem very similar as well. And if you look at the variances, they don't seem to be all that different either. So it's all the same. We should proceed and run an ACE model, right? Well, hold on. How do you know for certain? And the way you know for certain is by testing a saturated model. You'll do this test of a saturated model because when you look at the classical picture of an ACE model, you generally see something like this. And what you're seeing here is that the means of the weights for twin 1 and twin 2 are already assumed to be equal to one another. Similarly, the variances between twin 1 and twin 2 are also assumed to be the same in this model. Finally, the means and the variances for MZ twins and DZ twins are assumed to be equal to one another. And that makes intuitive sense. For example, we might consider it a bit odd if the mean weight for all twin 1 members of twin pairs were significantly higher than those of twin 2 members of the pair. It would give us pause and cause us to ask whether or not the assumptions that we are using to run an ACE model would be sufficient. And so if you are violating those assumptions and you still run an ACE model, you may be running the wrong model and you might get nonsensical results from your ACE model. Therefore, if tests of the data assumptions were violated, it would alert you to the need to carefully review how the data were collected and understand how these differences arise and whether there are opportunities to address these issues in the way you model your twin data. So running a saturated model and associated submodels to test your data assumptions prior to conducting an ACE model is highly suggested. Again, for this example, we're only focused on the single sex pairs, female pairs. So our situation does not need to consider sex. However, sex and the variance due to sex would be tested once we introduce male pairs or opposite sex twin pairs. And we're going to address this issue in a later session. Okay, to recap, up until this point, we have established one, that some expectations exist around the degree to which genetic and environmental factors will contribute to the variance of weight. And two, we have calculated summary statistics and also established some expectations around the equality of the means and variances to be able to test our expectations of that ACE model. So now let's turn our attention to the process by which we're going to conduct our work with the saturated model. First, we're going to test the saturated model and estimate the parameters that result from that saturated model. And what do we mean by the term saturated model? Well, a saturated model refers to a model that fits as many parameters to all the means, variances, and covariances as possible. And so once we run that saturated model, we'll take note of the model fit that results from that saturated model and compare it against three submodels. The first submodel is a model where you test the equality of the means of the twin members. The second submodel will build upon the first submodel, equating the means, and also test the equality of variances between twin members. The third submodel will build upon those models and also add a test of the equality of means and variances by zygosity. So if we were to go ahead and run our saturated model from our script, we would actually produce the results of the means, variances, and covariances as summarized in this table. If you compare the results from this table to the table that we previously reported from R, you would notice that the estimates are pretty similar. The other thing that I want to bring to your attention is that each one of these cells represents an estimated parameter. Therefore, we have 10 total parameters estimated in this model. And those parameters are estimated as the mean for twin 1 members of MZ pairs, the mean for twin 2 members of MZ pairs, the variance for twin 1 members of MZ pairs, the variance for twin 2 members of MZ pairs, and the covariance for MZ pairs. Those same 5 parameters that we've identified in the MZ pairs are then repeated for the DZ pairs. Another thing to note is that because we're able to calculate the covariance between members of MZ pairs and the covariance between members of DZ pairs, we can use that covariance estimate to calculate the correlation between members of MZ pairs and DZ pairs. So that's another nice thing that comes out of the saturated model. So now that we have a sense of all the parameters that are generated from a saturated model, and we have their estimates, we now want to revisit our understanding of the saturated model using a graphical representation via this path diagram of what a saturated twin model looks like. Here you have the variances for twin 1 members of MZ pairs, twin 2 members of MZ pairs, twin 1 members of DZ pairs, as well as twin 2 members of DZ pairs. Additionally, you have the covariance for MZ pairs, as well as the covariance for DZ pairs. And finally, you have a mean specific for twin 1 members of MZ pairs, a mean specific for twin 2 members of MZ pairs, another mean that's estimated for twin 1 members of DZ pairs, and finally, the mean for twin 2 members of DZ pairs. Now that we have run our saturated model and generated parameter estimates for the means, variances, and covariances, we can test all of the submodels that result after the saturated model. And we can produce a series of model fits for each of those submodels. And we can combine them in a model fit comparison table like the one that we've identified here. So I want to bring a couple of things to your attention. in this table. The first is the parameter estimate column. We just discussed that in the saturated model, there are 10 estimated parameters. Now, we would like to estimate a submodel where we make the means of the twin ones and the twin twos for an MZ pair to be equal to each other. And in that same model, we are estimating the means of twin ones and twin twos to be equal for DZ pairs. When you do that, you reduce the number of parameters from 10 to 8. Additionally, when you take a model now where you are equating the means of twin ones and twin twos, as well as the variances of twin ones and twin twos, you further reduce the model down to 6 estimated parameters. Finally, when you equate the means and variances for twin ones, twin twos, and between MZ and DZ pairs, you take down your number of parameters by another 2, and now you're down to an estimated number of parameters of 4. And so you note in this parameter estimate column, all of the actual number of parameters estimated in a specific model. It should also be noted that each one of the subsequent parameters where you'll be equating some feature of the saturated model are considered submodels of the saturated model, and they contain fewer numbers of parameters. Submodels will always have fewer numbers of parameters compared to their full model by which they're going to be compared against. Additionally, each model also has a measure of model fit as denoted by this column -2 LL, or -2 log likelihood. The -2 log likelihood is an indicator of model fit. And that model fit has an associated number of degrees of freedom as identified in this column. Further, these models will also estimate a measure of model parsimony to take into account some way of comparing these models while addressing the differences in the number of parameters across each of the models. So this measure of model parsimony is estimated using Akeiike's information criterion, or AIC, and those are listed in this column here. In both the situation where you are testing model fit as well as model parsimony, lower values indicate improved model fit. So lower is better when you compare model fits against one another. So how do you tell the difference between two models? What you're going to do is take the difference in model fit between one model, and you're going to subtract that value against the value of the full model. So in this case, when you subtract the value of the submodel against the full model or the saturated model, you will get a value of 0.22. And you do all the subtractions between each submodel and the saturated model. And you will also take the difference in the number of parameters to identify the difference in the degrees of freedom between two models. So in the second model, the difference between the number of parameters is 2, and the next one, the difference is 4. An interesting feature of this difference in model fits is that the value of the difference approximates a chi-square distribution. And so now you're able to calculate the p-value of this difference value as a chi-square with degrees of freedom that you've estimated as a difference in the number of parameters. And so this last column reflects the p-value of the test comparing a model that has fewer parameters against a model with more parameters. And so now that we've oriented ourselves to what is being summarized in this table and the general results from our code, let's consider how we are going to interpret these results. Here, what we see is that there are no significant differences between any of the submodels and the saturated model. Therefore, we can say that models where the means, variances, and covariances are equal by zygosity and between twin pairs, these models fit as well as a model with more parameters. Therefore, we can say that models where the means, variances, and covariances are equal by zygosity and twin pairs fit as well as the saturated model that has more parameters estimated. And therefore, we can use a simpler model to explain the data in a way that is as good as a model with more parameters. Further, these results indicate that we have not violated the assumptions of the ACE model. So now that we have tested our model fit assumptions, it is time to move on to actually doing an ACE model. And that's our last step here on the roadmap. We're going to estimate those genetic and environmental contributions and determine their magnitude of their contributions and the degree to which their contributions are significant using the ACE model and its related submodels. So when we look at an ACE model, we could use this path diagram and we would conclude that the variance of weight results from three sources. The variance due to additive genetic influences, the variance due to common environmental influences, and the variance due to unique environmental influences. These three sources represent three parameters that are estimated by the ACE model. An additional parameter is estimated as the mean of weight. It is also important to note that there are several assumptions of an ACE model, and they've been listed here. One of those assumptions, as we've discussed before, is that MZ twins are genetically identical. Another is the notion of equal environments, or that the environments twins are both exposed to, such as rearing environments, do not differ significantly between MZ pairs and DZ pairs. Another assumption is the presence of random mating, or the idea that people tend to fall in love and marry and have children in a completely random fashion. Additionally, we make the assumption as part of this ACE model that genetic and environmental influences are independent and function in an additive fashion. Therefore, the ACE model assumes that there are no influences on the population variance due to gene-environment correlation, gene-environment interaction, sex limitation, or the idea that genetic and environmental influences are contribute to the variance of the phenotype differently in men and women. There is no presence of gene H interaction and no variance due to epistasis. Although these mechanisms might influence the variation of this outcome, these are not the assumptions that we're holding for this ACE model. In future sessions, we will extend the ACE model to address some of these other sources of variation and address possible limitations of these assumptions. So if you go further down in the code, you will see that we actually run the ACE model and its related submodels. And if you were to do that, you would produce a table similar to this. And what you see in this table is very similar to what you saw in your saturated model table, an explanation of the model that's being tested, as well as a summary of the number of parameters that are estimated for each model, its related model fit, the measure of model parsimony for that particular model, as well as comparisons of model fit between a submodel against the ACE model. So for example, in the second row here, we're testing a submodel where we fix the estimate of C to be 0, or we remove the influence of C in this model, and we report an AE model. And when we test the difference in model fit between an AE model and an ACE model, we see that there's a significant difference in model fit. Additionally, when we look at the value of model fit against the AE model and the ACE model, we see that the AE model value is higher than that of the ACE model. Remember that lower values of model fit indicate better fit. So in this case, the fit of an AE model is significantly worse than that of an ACE model. We go on to test each of the other submodels under the ACE model, and we compare them against the ACE model. And in each instance, the ACE model is the best fitting model, the model that best describes the data. So we will proceed with reporting results from that model, and we have those estimates identified here. You'll notice these first three estimates, VA, VC, and VE, are the unstandardized parameter estimates for additive genetic, shared environmental, and unique environmental influences on the variance of weight. What you see here with SA, SC, and SE are the standardized estimates of A, C, and E, and the sum of their values will be equal to 1. So these values, these standardized estimates, are meant to estimate the proportion of the variance due to these sources. And what we can see so far is that the variance due to additive genetic effects contribute approximately 40% of the variance for weight. So this concludes the general concepts related to the roadmap for conducting twin analyses and thinking about twin data. And if you want, you can stop right here and take a break. However, if you're interested in understanding how we implemented the code to produce the results that we previously presented, keep watching, and we're going to get into a finer level of detail. So remembering that we had the saturated code and we used this path diagram to get us through. We're re-evaluating the path diagram and putting some color to it to help you see how this path diagram is implemented in OpenMx. So here we have one matrix that is called mean MZ, and that matrix contains one row and NTV columns. In this case, NTV has been generically defined previously in the code as the number 2. So this matrix has one row and 2 columns. Both of the cells of this matrix will be estimated as denoted by this term free equals true. And its starting values have been defined using the object SVME. This value, SVME, was also predefined in an earlier portion of your code. The 2 cells inside of the matrix have labels of MZ1 and MZ2 to denote the means for twin 1 and twin 2. So similarly, when we define the matrix in OpenMx called mean DZ, we have a matrix with one row and 2 columns. And the 2 cells that are inside of this matrix are labeled MDZ1 and MDZ2. All of the information that has been established in this matrix will then be passed into an object called mean DZ. So now we have identified and defined our means matrices. Now we're going to turn our attention to defining the matrices that contain the variances and covariances for MZ and DZ pairs. Here, we have a matrix that is going to contain the MZ variance and covariance, and it has been defined as a symmetric matrix with 2 rows and 2 columns, and all the cells will be estimated. Also, the starting values have been predefined using the values SVVA and NTV. Again, those values have been predefined previously in the code. In order to help OpenMx estimate accurately, we've placed some values of lower bounds as defined by this value val diag and LBVA by NTV. Those values were previously defined earlier in the script. We also have values that represent the variance of twin 1 and the variance of twin 2, as well as the covariances for twin 1 and twin 2. And because this matrix has been defined as symmetrics, we only need to identify the lower portion of the matrix. And the lower portion of the matrix has cell names of VMZ1 for the variance of twin 1 members of the pair, VMZ2 for the variance of twin 2 members of twin pairs, as well as the covariance between twin 1 and twin 2. The same approach has been used to define the variance and covariance matrix for DZ twin pairs, as we've identified here. Once we've identified all those matrices, it's time for us to put them together and actually put them to work in an OpenMx model. And here, we're running additional code where we identify the data using the mx data function, and we're telling it which object contains the data, as well as the type of the data, which is raw data. We have also identified several additional pieces for the models using matrix names, as well as the object names for all these groups. And we put those in the models, and we run these models using the mx run statement. Put in those models, and we run the models using the mx run function over here. And everything that's produced from this model is passed to an object called fitsat. If you want to look at the results that have been produced from that model, you can start by looking at a summary of the object fitsat using the summary fitsat function. And then you can dig even deeper into that fitsat object and pull out all kinds of other more detailed results from that object. Okay, so this gets you a sense of how we ran the saturated model, all the steps that went into running that saturated model as well. But we also ran submodels where we were equating the means and the variances. Here we're going to show you one example of one submodel where we equated the means across members of twin pairs. And in this case, we used the model fitsat that we had just run from the saturated model, and we apply it to the submodel. And we tell OpenMx the following: hey, OpenMx, I already ran this model fitsat that I want you to take all the matrices that we used in that situation and all of the matrix definitions. And I also want you to take all those algebra definitions from there, and I want you to apply them and place them into a new model called 1EMOC. And I want you to have all that information of that model passed to an object called model EMO. The nice thing about doing this approach is that you don't have to rerun a new model completely from scratch. It is holding all of the information, all the matrix definitions from your saturated model, and applying them now to your new, smaller submodel with fewer parameters. So once you have that, and you are now moving and working with this new model, EMO, you'll still need to tweak it a little bit more. And you say now, OpenMx, I want you to look for the labels that were previously listed as MMZ1 and MMZ2. And I want you to put the label MMZ into each of these cells. The fact that you have now used new label names tells OpenMx that you need these parameters to be equated. That is our way of defining to OpenMx that we're equating the means for the MZ pairs in twin 1 members and twin 2 members. Similarly, we would do the same procedure for DZ pairs and changing the specific names of MDZ1 and MDZ2 to a single name, MDZ, that will be present in both of the cells of that matrix. We conduct this equating process using another function called OMX set parameters. So now we transition to providing detail on how we conduct the ACE model in OpenMx. This figure shows the same figure we've been using before, but right this moment, we're going to focus on the means portion of this model. In the code that we have identified here, we have defined a matrix to have 1 row and 2 columns, and both of those cells in that matrix will be estimated, and both of them will have starting values of SVME. Additionally, we have labels of lab bars, and each of the twin members from this cell will have the value mean estimated. So in your 1x2 matrix, each of those cells is going to have a value mean identified. And this whole matrix is called mean G. And everything of this mean G matrix is passed into an object called mean G. So now that we've identified the matrices for the means, let's talk about the matrices that are holding the information for VA, VC, and VE. Each one of these matrices are 1x1 matrices, and their values will be estimated. Additionally, the matrix information that has been identified here is then passed into objects COV-A, COV-C, and COV-E. We also have variance and covariance matrices, which we will produce using matrix algebra. So we take the matrices of VA plus VC plus VE, and we add them to estimate the total phenotypic variance. The result from that addition of those 3 matrices resides in this matrix that's called V. And all of that information is passed into an object called COV-P. We are also calculating the covariances of MZ pairs, which is estimated as VA plus VC. Additionally, we are also estimating the covariance of DZ pairs, which is 0.5 VA plus VC. And each of those covariance estimates have been identified here and here. So now we are also identifying the matrices that need to be utilized for the estimation of these models and the objects in which the matrix information is contained as defined in the MX model statement here. Finally, we identify a fit function by which to conduct the model. And we do one last piece of algebra where we are estimating the unstandardized and standardized estimates. And here we do one last piece of algebra where we report the unstandardized A, C, and E estimates and also calculate the standardized variance components estimates identified here as VA divided by V, VC divided by V, and VE divided by E. And all of the algebra results are contained within this object STUS. Finally, we run the model and pass the model that has been run with the ACE model information to an object called fit ACE. Then when we want to look at the basic summary of some of that ACE model, we would take a look at summary fit ACE or the object sum ACE. And as always, if you want to take a deeper dive into the results of those models, you simply need to open up and work with the object fit ACE. So with that, we're going to stop for today. And if you have any questions, please reach out.